It's live streamed.
I guess we can start bringing people in.
Oh, they're already coming in. Yeah, there's already 30 people and more.
Sure, but I mean promoting them.
Oh, that for sure. Yes.
So there is a bunch of people, I'm guessing, from your neck of the woods, Mohit. I don't know who some of these people are, so but
Well,
we can.
It's open for everyone, right?
Yes, I'm just saying, but in terms of promoting them to
I see. Yes.
a panelist.
Mohit, as I said, if you want to share your screen, that's okay.
Sure, I'll just share my screen.
Because as we make some of these people panelists, that means that the screen will get smaller and smaller with faces, and really the focus of the presentation is going to be you and your presentation. Cool. I think I'm going to start introducing you, Mohit, so you have as much time as possible to give your presentation. So it's a distinct pleasure today to be introducing Dr. Mohit Kumar Jolly, who I like to say is a person who doesn't need an introduction in this community. That's usually something that you keep saying about people who have been in the field for many decades, and Mohit is obviously a lot younger than that, but he has had quite a lot of impact in the community recently in terms of not only his publication record, but also he's a co-leader for the Math-Onc subgroup at SMB. So clearly he's someone whose impact both in India and the United States and elsewhere has been easy to see. So Mohit got a degree in the Indian Institute of Technology at Kanpur, also a master's degree before he moved on to the United States to do a PhD under the supervision of Herbert Levin at Rice University in Texas, where he also stayed on to do his first postdoc. And then he moved back to India, where he's now an assistant professor at the Indian Institute of Science in Bangalore. So he is definitely one of the leaders in the field of math oncology, especially in the subfield of plasticity and epithelial-mesenchymal transitions. I think that you actually had to compile in 2 volumes all the publications in plasticity in math onco. I think you can have 1 volume just for Mohit and 1 for the rest. Just that's how much productivity he's got. I challenge you to go to Google Scholar to find out his publications, because it's quite a lot in many good journals. So he'll probably have to make an effort to squeeze a presentation in the space of time that we have for him. So Mohit, please, stage is yours. I don't think we can hear you. Are you? Yes, you're muted. Sorry, Mohit, you're muted still.
Sorry, my bad. Sorry. Can you hear me now, right?
Yes, it's perfect now. Awesome.
Fine. Thank you so much, David, for that very, very kind introduction and for the kind invitation to share some of our work here at this seminar series. It's really an honor to be presenting some of our work here. I've really been following much of the exciting work and have been quite a bit motivated and encouraged by work coming out from IMO for quite some years now. So it's a real privilege to present some of our work and get critical feedback as well on some of these ideas that we have been working on. So today I will talk about some of the concepts that we have been investigating from a dynamical systems perspective, looking into trying to construct the landscape of epithelial-mesenchymal plasticity as it plays out in the context of metastasis, which, as most of you know, still remain the major cause of all cancer-related deaths. Now, what attracted me from a dynamical systems perspective to look into this question was that metastasis is actually a highly inefficient process, and it's very small percentage of cells that are eventually able to form secondary tumors at distant organs after they leave the primary tumor. And this is not surprising because a cell that is en route metastasis has constantly changing environment around itself, and with those changes come different challenges that cells need to adapt to it at a relatively fast time scale, which is not commensurate with that of cell division. So therefore, these adaptations need to happen at a much faster time scale and need to happen at a phenotypic level rather than a genetic level. And because these different challenges that are being posed by different elements that are shown representative in this particular cartoon, cell needs to adapt to many of them together. We also need to identify and better understand how this adaptation happens on multiple axes simultaneously. And is this also one of the reasons why metastasis happens to be such an inefficient process? So this idea of cell state switching or phenotypes changing in a reversible manner has been investigated in the context of developmental biology for quite some time, where, for instance, people have been developing these landscapes based on gene expression profiles and talking about how cells can switch back and forth under the influence of noise. And the implications of this can be far-reaching in the context of cancer as well. Because, for instance, as shown in this particular example, let's say if you have a cell population and you look at a particular molecule of interest and sort out these cells separately after facts, what you are likely to observe is that even one subpopulation has the capacity to give rise to another over a certain specific duration based on the transition rates that can be calculated based on such landscapes. So this ability to switch back and forth can also drive this heterogeneity at a phenotypic level or at a non-genetic level, which is getting increasingly recognized in the field of cancer metastasis and drug resistance as well. So this was an example from developmental biology, but this has been investigated in cancer cell lines as well. For instance, in this one of the landmark studies, it was observed that a given cell population has 3 different states based on different cell surface markers. And when you segregate all of them and wait for the system to equilibrate, they returned back to the parental distribution, indicating more like a Markov kind of process in this scenario. We have, in collaboration with our experimental colleagues here, also done these experiments where you would sort out these cells based on a given molecule of interest in this particular case. And again, we saw that this repopulation kinetics was clearly seen irrespective of where you start with. So this seems to be a more generic property rather than a very context-specific scenario. So the questions of our interest sort of rotate around the theme of cellular plasticity, which, as we were just talking about, can come in various different flavors, such as the ability to resist drugs, such as the ability to evade the immune system, the ability to change the proliferation status back and forth, and so on. And its importance is being increasingly recognized in the cancer biology literature. And in the latest edition of the Hallmarks of Cancer, this plasticity and consequent non-mutational reprogramming were included as these 2 enabling hallmarks. So the questions that we are interested in trying to address is how many states can cancer cells exist in? How do they switch among these states? What networks enable these switching? Is it always just cell autonomous, or does it also depend on the environment? Does it also depend on the population distribution itself? Is that more like a quorum sensing kind of scenario that is going on? And of course, how do they coordinate behavior among these different axes? So when they're changing their behavior, let's say their metabolism from oxidative phosphorylation to glycolysis, are they also changing their ability to evade drugs or evade the immune system, and vice versa? So how are these different seemingly independent axes connected to each other? How does one knob turn on or turn off any other engine that exists on seemingly independent axes? So, of course, there has been a lot of work more recently in collecting this kind of data that can show a static snapshot of how this heterogeneity looks like. The dynamic data has been difficult to collect for various different reasons. And what we tend to do is sort of look at these static snapshots from different patients, from different cell lines, and try to understand where is this layers of heterogeneity coming from, and what implications can this have in the context of metastasis and drug resistance? And eventually, a more fundamental question, can we even think about controlling this emergence of this heterogeneity and the implications that this has in a system that is as dynamic and as adaptive as cancer itself? So broadly speaking, our group sort of works on 3 sets of questions. One is one set of team members look into specific networks, identifying in different cancers or in different axes of cancer plasticity, what networks enable this ability to switch back and forth and therefore give rise to multiple states. Another set of team members work on why are networks designed the way they are? Why has evolution chosen a particular strategy as compared to a million other possibilities that could have been there? And then the other set of team members look into sort of zooming out from what is going on within a cell to its implications in terms of what's going on at a tissue level behavior and at length scales and time scales, which are much larger than what typically an individual cell plasticity is consistent with. And we are able to look into these questions because of the plasticity and heterogeneity that exists in the lab from students coming from all different backgrounds, brainstorming ideas and thinking about the same process, but from very different perspectives. And of course, our collaborators who again come across different disciplines and have played an instrumental role and continue to do so in shaping some of our analysis. So I would like to acknowledge them right up front. So today I'll be focusing on this specific axis of epithelial to mesenchymal transition, which has been very well studied at a molecular level for quite some time now. The general idea here is that if you think about carcinomas, which are epithelial cancers, so those cancer cells typically do not have the property to start migrating or invading. Rather, they are sitting forming very tight cell-cell adhesion bonds with their neighbors. And when it comes to metastasis, they usually shed some of these properties and on the other hand, gain the properties to migrate and invade. And this transition is what is called as epithelial to mesenchymal transition, which is postulated to be the first step in the process of metastasis when cells are leaving the primary tumor. And once they reach the other organ, they are thought to undergo this exact opposite transition where they regain the ability to adhere and lose the ability to migrate or invade because they no longer need to. They actually need to settle down and form a macrometastasis or a secondary tumor. And there, the exact opposite transition, MET, is believed to play an important role. And these cycles of EMT and MET, which again are not unique to cancer, they happen in developmental biology and wound healing as well. So these cycles are thought of to be playing an important role in enabling multiple rounds of metastasis. These are some snapshots of representative figures which taken from the field of EMT to almost 2 decades ago now. And what you see in all these snapshots sort of conveys what the field has been thinking about the process of EMT, that cells would convert to being mesenchymal, and once they reach the other organ, they would convert back to being epithelial. If one sort of looks more carefully at these, there are various different assumptions that are hidden in these cartoon models here. And those assumptions, some of those assumptions are that EMT is thought of as a binary process. Cells have either epithelial state or a mesenchymal state. It's thought of as a reversible process, that cells which undergo EMT come back to being epithelial. And it's also has been assumed to be a symmetric process, that properties that cells lose while undergoing EMT are thought to be regained as they undergo MET or vice versa. So these are some assumptions that we started asking questions about using mathematical modeling and going back and forth between experimental data and predictions being made by mathematical models. So just to give a snapshot, this is the first model that we made for EMT network almost 10 years ago now, where we collated information on what different molecules were known to regulate a more epithelial versus a more mesenchymal phenotype. And this is the sort of simplest gene regulatory network we could come up at that point of time. And here also there are various assumptions, like multiple molecules belonging to the same family have been clubbed into as one node and so on. The numbers represent the number of binding sites and some quantitative input-output relationship that can be drawn based on that. And there is transcriptional regulation as well as translational regulation that is sort of going on. So here, these two molecules, which are both transcription factors that are driving a more mesenchymal phenotype, and here these are two microRNAs which are driving a more epithelial phenotype. And what you see between them is this mutual inhibition. So it's like a tug of war that is going on, which is trying to pull the cell towards either an epithelial or a mesenchymal cell fate, depending on the relative levels of these two sets of molecules that you see here. Now, of course, these kinds of investigations have been there in bacterial setup, in bacterial systems, and also other mammalian systems as well. So what our analysis here, our focus here was that can we now write down a set of mathematical equations based on these interactions and see what the model predicts. So we came up with a simple...
Can I ask a question?
Please, please.
So, I mean, it's just more for probably the benefit of the greater audience. I'm sure everybody knows about the epithelial-mesenchymal transition and what it means, but maybe fewer have thought about the idea of it reversing, right? You know, that going from mesenchymal to epithelial. And so I guess from a kind of, let's say, functional point of view, the idea is that an epithelial cell can turn into a mesenchymal cell through this process, right? It's not just that it becomes migratory, it can actually turn into some sort of, you know, eventually a stromal cell of sorts, right? Is that right?
Yes, so it begins to express certain molecules, you're absolutely right, which are often expressed, say, only by fibroblasts, and begins to show certain properties such as contractile properties, which are often associated with stromal cells alone and not with the epithelial cells.
I guess the key point here is it actually turning into a fibroblast, or is it just expressing the sort of markers and behaviors of a fibroblast? Do you see what I'm saying? Is it actually becoming a fibroblast, or is it acting like one?
I think it's more like acting like one rather than actually becoming one.
Right.
So it's more fibroblast-like rather than a fibroblast itself.
Because obviously there is fibroblasts can come through other means. And so I guess, you know, from that point of view, then the reversal isn't so shocking if it's just sort of like pretending to be a fibroblast, that it could then come back to being an epithelial cell, right? But for a long time, it was thought that that was a one-way process.
Exactly, yes. So that was, I think, in early 2000s when it was still thought of as a... So EMT was initially called, you're very right, so thanks for pointing that out, Sandy, epithelial to mesenchymal transformation, when it was thought that this is a one-way process and this plays an important role in cancer initiation itself. But then later it got replaced with epithelial to mesenchymal transition, when people realized that it's more of a reversible process and it's not really directly connected with the malignant transformation or immortalization itself.
Yeah. But it's more for the rest of the audience. And I think another important point there is that then realizing that it's not binary, it means also there's intermediate states, right? And I presume you're going to talk about that.
Yes, exactly. That's what I was saying.
Thanks.
Thanks, Sam. So, so at that point, we sort of basically looked into this framework where one or more microRNAs can bind reversibly to an mRNA, forming these complexes, which can then lead to degradation of mRNA or just transient sequestration of these. So this is straightforward. You can convert this into a set of coupled ODEs where every variable has one equation corresponding to it, and you have terms of production, degradation, microRNA regulation, and transcription regulation. So long story short, what our model predicted was, as Sandy was rightly pointing out, is that it's not a binary process. You can have, under some parameter combinations, a third stable state that cells can attain. So here in this bifurcation diagram, the blue curves show the stable states and the red curves show the unstable states. So what you see here is that there's an epithelial state with very low levels of Z, a mesenchymal state with very high levels of Z, and in between that there is a third state which you can think of it as a hybrid epithelial mesenchymal kind of state, and also has properties which sort of combine the traits of epithelial and mesenchymal. So adhesion and migration put together, possibly giving rise to collective cell migration, which is distinct from individual migration that is seen in a mesenchymal state. So that's about 3 different states. And as you can also see in certain parameter regions, these states can coexist. So in this region, the only one state is epithelial, if you draw a line vertically upwards. Here, again, the only state that is available is mesenchymal, but there are regions in which more than one state is available to a given cell. So what that means is that, of course, EMT is not binary, and under the influence of biological noise, it is possible that cells can switch back and forth between these states even when they are not receiving any external signal. So that's the property of the system itself. Those were certain predictions that we had made at that point of time, and we collaborated with different colleagues to test out these predictions. So in this particular case, these are non-small cell lung cancer cells stained in red and green for E-cadherin and dimension, which are canonical epithelial and mesenchymal markers. And what you see here in these images is that individual cells do express both red and green, suggesting that this in vitro model system can be taken as a proxy to investigate the hybrid phenotype further. And the fact that this phenotype can be maintained over multiple passages in the lab indicates that it's a stable system and not just a spontaneous, always transitioning to other states as well. So then we wanted to further quantify this, and we used the same two different molecules that I was just telling you about. And we quantified the number of cells which are dimension positive, E-cadherin negative, E-cadherin positive, dimension negative, and double positive in breast cancer, prostate cancer, lung cancer, and colorectal cancer cell lines. And again, you can see that these cell lines which have identical genetic background, you do see phenotypic heterogeneity patterns here. And finally, this was another study which was done independently by another group, where they segregated these three different subpopulations and they cultured them independently, and noticed that each population was, each homogeneous population was eventually able to give rise to a heterogeneous population because they showed the propensity to switch. And of course, the rates of switching are not identical. They depend on which direction you are switching and where you are starting from. And this is also something we have worked on, but I'll not be able to talk about it today in the interest of time, but happy to chat separately. But all these three different predictions we were able to sort of see across different scenarios. And once we made these predictions in the past 10 years, there has been a large number of papers which have modeled different EMT networks. Each of them has focused on a different set of nodes, depending on possibly a specific cancer, depending on certain pathways of interest, and so on. And they have predicted, for instance, in this example shown here, that not only one, but actually more than one intermediate states can exist. And again, the larger the network you look at, the more the number of these hybrid states that you can get. In fact, in this latest paper, where they used the Boolean models instead of ordinary differential equations, they were able to see many, many hybrid states. And they also saw that there was a power law distribution in terms of the probability of distribution of this steady state itself. So clearly saying that yes, EMT is not binary, and this prediction seems to be validated across different models and across different experimental data as well. So that's fine.
Quick question. Sorry.
Yes.
So the on your previous slide, but it's also applicable to this too. In terms of the stability of these states, according to your model, is that sort of transition state or the middle state unstable? Or is
it is the least state. It is the least stable state. Yes.
Is okay.
As compared to the other two. Yeah. Thanks for pointing out. I'll come towards that point in the end as well in a more quantitative manner. Yes. So hybrid states exist. That's fine. But what are its implications in the context of metastasis? So again, one of the predictions we had made by looking at a network which includes the EMT players as well as some of the other players known to be involved in tumor initiation potential, or stemness, in other words, we predicted that the hybrid state is actually the most stem-like or the most tumor initiating, perhaps the most fit for the purpose of metastasis as compared to epithelial as well as mesenchymal ones. And there has been more and more data across different cancers which has been coming out now showing that indeed that prediction is true. There was a recent paper which claimed that hybrid states were actually essential to form breast cancer metastasis. And in this particular case, from squamous cell carcinoma, they identified cells in multiple states of EMT. So you can see multiple intermediate states here. And they showed that the ones which were most metastatic were not the ones which are completely epithelial or completely mesenchymal, but the ones which are somewhere in between. So tumor initiation potential as a function of EMT happens to be a non-monotonic response, which initially increases and then sort of settles down again. So these predictions have been again tested in multiple different systems now, and now the conceptualization of EMT, even the terminology has now changed. People begin to talk about epithelial-mesenchymal plasticity to identify or to emphasize that there are multiple intermediate states that are possible. How many states exist? What is their characterization is still a very active area of research, which of course depends on which cancer you're looking at and whether it's an in vitro or in vivo system as well. So coming to the next question, is EMT a reversible process? So you would think from this representative diagram that the EMT engine has to be switched off so that cells can regain proliferation and eventually metastasize. So we wanted to look at some studies which had data on whether EMT was always reversible. So there were these couple of papers which focused on some tipping point kind of behavior. So some of the experiments that were done in these two papers were that you would drive EMT using TGF-beta, which is one of the most common EMT inducers established in vitro. And you would see whether the cells come back or the cells stay mesenchymal after you wait for a specific period of time. But there was no understanding of what is causing this and whether this tipping point, at what time point is this likely to exist. So we again made a very simple phenomenological models without going into too much details of what is happening at a molecular level. We said that because many of these molecules such as Z1 and other transcription factors are capable of remodeling the chromatin in such a way that even when their expression goes down, the chromatin has been remodeled and the effect of Z1 will stay longer in a cell even when Z1 has gone down. So there's a memory in the system, there's a resistance in the system that is being built up, which is likely to be a direct function of how long a particular gene has been on. So this was adapted from a model. Here what you see is that now the threshold function that is often seen in such ODEs is no longer a constant variable, but it actually depends on how long a particular gene has been on. And the stronger this epigenetic feedback is, the lower this threshold and the stronger the impact. So we took the network that I've been showing you earlier and said that what happens if I now include such an epigenetic feedback from ZEB1, which is a potent chromatin regulator, in terms of inhibiting microRNA 200. So when we do this, so all these diagrams are increasing levels of epigenetic feedback. What you see is that these diagrams are changing. And if you look at the population distribution, so without any feedback, if you look at the population distribution, you would see approximately 80% cells for this parameter region are epithelial in nature. But now if you include epigenetic feedback, you see that the population distribution changes and you have only 40% of cells which are epithelial and many cells are actually mesenchymal. So what this tells us is that EMT can be irreversible if the chromatin mediated changes have happened to such an extent that it is difficult to get out of that state. And this is again not only true for EMT, even MET can be irreversible. So you can have cells, no matter what amount of TGF-beta you give to it, those will just not exit the epithelial state because they're epigenetically locked into that state. And again, there were some observations similar to this made at a bulk level earlier as well, saying that EMT is actually a rare event. It's a very small percentage of cells that undergo EMT, but more recently, single cell investigations have also helped us to understand where this resistance to EMT or resistance to MET more carefully, and also its association with the chromatin changes that often happen as a function of that. So for instance, in this scenario, single cell clones were established from a given cell line. You induce EMT, some of them downregulate epithelial, upregulate mesenchymal, and they don't come back to where they started from, as you can see. And in some of the cases, they just don't really downregulate their epithelial, and they are able to come back. So we still need to better understand what chromatin regulators are being used and so on. But again, as a proof of concept, there is enough evidence now, at least in vitro, that EMT can be irreversible as well.
Can I ask a question? Sorry, Mohit,
yes,
on the model side before this.
Yeah.
So I didn't fully understand what the epigenetic feedback, what does that mean here in this model? What is it you're actually changing?
So we are changing the value of alpha here. This is a variable that you can alter.
Yeah, but what does it represent in the greater model?
Yes, so it represents, so if, as you can see, if the value of alpha is higher, then this particular value, which is the threshold in the Hill's function, would be smaller. And the threshold smaller means that now you can inhibit MIR200 even with a smaller value of ZEB1 than what was earlier required.
So it's kind of making this, it's amplifying the impact of the signal in some sense.
Exactly. It's amplifying, okay. Yes, in terms of time as well. Yes,
yeah.
Not just instantaneously, but it's sort of keeping a count, because this is a derivative here. And as a result of which that impact is relatively long-lived.
Okay. Mohit, can I have a quick question about this? So this epigenetic locking or epigenetic feedback, is this happening on the single cell level, or how the cell communicate to make this collective decision?
That's an excellent question. We don't know whether in a given population of cells, what percentage of cells are undergoing epigenetic locking and to what extent. That data, to some extent, it's there in this recent preprint where they've established clones out of single cells from the same cell line, and then showed that there is a difference in the chromatin landscape. But I think they also sort of fall short in establishing a causal connection. All they show is that there are different chromatin landscapes and there are different levels of resistance. But are those chromatin landscape essential to give rise to that resistance or locking is something that needs to be looked at more carefully.
Thank you.
Thank you. So the other question which we had was, is EMT a symmetric process? Now, as you can look into this bifurcation diagram, the path that the cells are taking to undergo transition from E to M and the path that they are taking from coming back from M to E is not the same. There's hysteresis, which is clearly seen in the system. So suggesting that it is possible that cells do not, even if they come back, they might not come back to the same gene expression profile that they started with. And there was this recent investigation in prostate cancer cells where they treated cells using 3 different independent inducers of EMT. And then, so this is the inducer, they induced for 5 days. And then they looked at what the gene expression profile looked like at a population level, 3 days after removal, 5 days after removal, and 20 days after removal. So you can clearly see there are certain genes which, once they go down, they don't really come back. So there is some level of hysteresis or irreversibility that is there in certain sets of genes. Now, how do those genes connect to this particular set of molecules shown here in this bifurcation diagram is something that we are still working on, but at least at a conceptual level, this was seen. And this was seen not only at an transcriptomic level, but more recently also in proteomics data, where based on mass cytometry in this particular non-small cell lung cancer cell line, EMT was induced for 10 days, and then the signal was withdrawn for 2 days, 6 days, and 10 days. And again, there was hysteresis which was seen because the cells did not follow the same trajectory what they had followed by going from epithelial to mesenchymal aspects. So sort of summarizing this part, what we started with is EMT a binary process? And the answer to that, driven by mathematical models and now validated by multiple different preclinical and clinical observations, is that clearly it's not a binary process. You can have multiple states which can even play a crucial role in the context of metastasis. It's not always reversible as well, and it's not necessarily always symmetric as well. Now, what parameters, what molecules regulate these reversibility and symmetry in the dynamics is, again, a very active area of research ongoing. So the next set of
before you jump on, because that's a great point to sort of let people ask questions. So, you know, from our point of view, it's very convincing, the story you're telling, and you're supporting with lots of evidence. And I guess one of the questions is, why would, in development, we actually build this process into the cells? Like, why would we have that there? Because it must be there for a reason, right? It must be needed, so to speak. Do we understand that?
That's a very good question, Sandy. So the reversibility, to some extent, we understand that, because these layers or multiple rounds of EMT and MET which are important in forming different organs. So say different layers of cardiac tissue require different number of rounds of EMT and MET, and also possibly starting from different cell types. So neural crest migration is one of the most well-studied examples of EMT and MET in development. And there you need cells to migrate collectively. So the terminal end bud cells which are there at the branch, they are supposed to be showing some properties of leader cells that they can actually mechanically pull the the follower cells with them. So that those cells have been now shown to co-express epithelial and mesenchymal markers, at least transiently. So clearly there are transitions in and out, and in the development process, this is very well spatially coordinated as well. That is something which we have not really looked at very closely yet.
Yeah. Because obviously understanding that could give insights into why cancer then exploits this somehow, right?
Yes.
And from that point of view, the hysteresis aspect of it is intriguing, because it might imply that, in fact, it takes more effort to go back than it does to go forward. And so, you know, maybe there's a way to exploit that somehow. I don't know.
That's a good point. I think the hysteresis aspects, in one of the recent studies, they actually, using CRISPR-Cas9, broke the feedback loop and then observed that the multi-stability was gone. And if you induce with TGF-beta, what you saw was a more gradual transition than a more discrete transition. So some of that analysis is coming, and more importantly, they showed that when you break that feedback loop, metastasis goes down. So if you're getting rid of multi-stability, if you're not allowing them to switch back and forth, then that can clearly have an impact in terms of its metastatic fitness. But how can we use it in other ways? That's a great point. That is something that I have not really thought about.
And I guess if we need this process, is it we only need it during, you know, embryo development, or do we actually need it as a day-to-day, like in terms of, say, organ turnover, repair, or whatever? Or is it we never need it now, and cancer just exploits this because it's like a hidden, so to speak?
Right. And in development and wound healing, it's mostly only a partial transition. Cells rarely undergo a complete transition, because when there is wound healing, you see collective cell migration and not sort of single cells flying out. So there is increased appreciation of the partial state in development and wound healing, even more than what is there in the context of cancer.
Thanks. I think somebody had a question in the chat. Let's see. Is there a way to artificially induce epigenetic locking in order to lock the cell state?
Yes, so if we know which knob to press, then yes, it is possible to do that. But we are not at that level of resolution yet in terms of a modeling. But there have been recent studies which are trying to map the ATAC-seq and RNA-seq simultaneously at a single cell level. So perhaps those kinds of multi-omic investigations can give us a better direction as to how to do that. But that's a great point. And there was a recent study where they pushed mesenchymal cells to become adipocytes by upregulation of PPAR gamma, and that was sufficient apparently to reduce metastasis again. So there are scenarios where people are trying to identify, just as in the context of cellular reprogramming, what knobs to push and to what extent, or what combination of knobs to push and to what extent to push these cells in to less aggressive forms. And one of those less aggressive forms is reduced plasticity, but there can be many others as well. Okay, so I'll move forward into some of the other aspects we've been looking at, is how do these hybrid cells? So far, we have been only talking about EMT. But what about other axes of plasticity? Does cells undergoing EMT also change those axes of plasticity simultaneously? And again, this is a question that Sandy earlier pointed out. Why hybrid cells are less stable or more plastic than epithelial and mesenchymal? And this is a feature which has been seen across different networks, across different modeling strategies, no matter which system that you're looking at. So is there something more deep in the context of how the network itself is laid out? So in the context of metabolism, what we did, we don't have a regulatory network at this point. We started with a data analysis kind of approach done by Srinath. What we did here is we looked at approximately 200 data sets in vitro, in vivo, across different cancers combined, and asked this question, whether the enrichment of 3 different axes of metabolism, oxidative phosphorylation, fatty acid oxidation, and glycolysis, how do they connect to enrichment of EMT? This is all gene expression based, gene set based enrichments. So what you see in one of these plots, the x-axis is the correlation coefficient. Each dot is a data set, and the y-axis is just telling you the statistical significant cases. So what you see here, for instance, is that in 56 out of 80 data sets, we see that oxidative phosphorylation is negatively connected to EMT. And in 24 cases, it's positively connected to EMT. So it's clearly there is a bias, and there's no one answer here. We are talking about seeing an asymmetry in this, because if there was no connection, then you would have seen equal numbers or close to equal numbers here, which is clearly not seen irrespective of which axis you look at. So what this tells us is that oxidative phosphorylation and fatty acid oxidation are negatively connected to EMT. So when cells undergo EMT, these decrease, but glycolysis increases. So then this is looking at one dimension at a time. Then you wanted to look at 2 of them at a time. So what if you take the corresponding data sets here and see what's going on? So again, the most common pattern is what you would expect here, that both fatty acid oxidation and oxidative phosphorylation are reducing as cells are undergoing EMT. That's why you see them in the 4th quadrant. But that's not the only solution. You do see cases in which they are positively correlated as well. So then we started to do more combinatorial kind of outputs and see which of them actually has the ability to predict survival in Cancer Genome Atlas TCGA data. So what you see here is now we have 3 different axes: EMT enriched or not, glycolysis enriched or not, oxidative phosphorylation enriched or not. So you have 8 possible combinations. So we start with a case where none of them is enriched, and then see which combination is able to predict poor survival. So the stars, the asterisks are the one where you do see a statistically significant trend in the context of poor survival. So when do you get poorer survival? When do you get worse survival? Is when the cells are more EMT and more glycolysis, but the oxidative phosphorylation can be positive or negative, suggesting that when cells are undergoing EMT, you need them to undergo glycolysis simultaneously, but there seems to be some degree of flexibility, some degree of freedom, whether simultaneously they're changing their OXPHOS or not. So again, glycolysis and OXPHOS are thought of as antagonistic processes. And what here we are saying is that it actually does not matter a whole lot. As long as their glycolysis has changed, you can get worse survival in this particular scenario. So we are still working on developing a more mechanistic model to understand these kinds of observations that we have had so far. So in terms of immune evasion, there are again reports on how activation of EMT can actually prevent cells from attack by immune system. And there are various different levels at which this can happen. There have been some fantastic mathematical modeling as well as experimental papers to look into this tumor immune population level dynamics. And what we looked at is what's going on within a tumor cell which is undergoing EMT, not really focusing on the population level dynamics there. So again, we had a simple network. Many of these molecules you might already be reminiscing from the previous slides, mid-200 ZEB, and these are the EMT ones, and here is PDL1. So PDL1 levels higher means the cell is more immunosuppressive. The T cell will not be able to attack. So what we see here is that if you take this network and simulate these ODEs over an ensemble of parameter sets taken from biological literature, what you see is that the behavior of PDL1 as a function of EMT is more like a saturating curve. So when you compare the PDL1 levels in epithelial versus hybrid, in hybrid, they are sufficiently high and close enough to mesenchymal and sufficiently different from what you see in epithelial. Again, suggesting that cells need not undergo a complete EMT to have immune evasion. Even if they undergo a partial EMT and get into this hybrid state, that is capable of evading the immune system, at least as far as PD-L1 levels are concerned. So then we looked at these predictions from multiple different experimental data sets where EMT was induced in different scenarios, and you do see changes corresponding to EMT players going up and PD-L1 going up. When you induce MET, you see PD-L1 comes down, so it's a reversible switch. And again, in this data set, which I talked about earlier, when EMT is induced, you see that EMT expression as well as PD-L1 expression goes up. And when they come back, it's again showing a hysteresis pattern. They're not coming back to where they started from. So this can have implications in terms of metastasis also being differentially sensitive to the immunotherapies that we have been talking about. So this is all at a bulk level. We wanted to see what's going on at a single cell level. So here was this data set where the 3 different cell lines were induced with TGF-beta and other regulators as well. And what we did here is we deconvoluted EMT into 2 axes, loss of epithelial and gain of mesenchymal. We often assume that they are happening simultaneously, but they need not be. So what if you calculate epithelial and mesenchymal scores separately? So for instance, here, this is 0 day, then 7 day of TGF-beta, and then 3 day removal after the treatment of 7 day. So what you see is that in this particular case, the epithelial score decreases, which is the x-axis, and mesenchymal score increases. So this is the typical EMT that is happening. But when you withdraw the signal, the change is only happening in the epithelial one. The mesenchymal score is not decreasing. And that sufficiently has high levels of PD-L1. And this was, again, same pattern was seen when you change only along the mesenchymal axis, but not along the epithelial axis. But if you don't change along any of the axes, then PD-L1 levels do not change. So again, what this is telling us is that if you lose epithelial or you gain mesenchymal, either of them might actually be sufficient to increase the levels of PD-L1, and you need not necessarily do both of them and undergo a complete EMT to gain the properties of immune evasion. So next instance is about drug resistance. So again, here we had to be specific as to which cancer we're talking about.
Can I ask a quick question? So
please.
This is so much data. I'm trying to take it all in, which is great. But can you just clarify, is this from cells in culture, in 3D culture, in 2D culture? Like how is this data generated?
This is a 2D culture in vitro induction of the EMT by TGF-beta of 3 different cell lines.
And so you're looking at that kind of hybrid state.
Each dot is a single cell. Yes, sorry, I should have explained that.
So what was intriguing to me was the piece that you previously showed, which was that basically the metabolic behavior of the cell also influences that state. And so is that in terms of the internal metabolic state, or what about the environment that's causing that internal metabolic change? Because obviously metabolism is something that's quite plastic, right? The cells will utilize the resources they can to survive. So if you put them in like a high glucose environment, does that drive up this sort of, intermediate state and make them express more PD-L1, for example, right? Or is it the cell actually has to change somehow metabolically for that behavior to occur? Just, I mean, is it the chicken or the egg?
Yes. No, that's a great question. So far we have looked at sort of only keeping EMT as the center and then seeing how different axes are changing. But that's the next thing we are now going to look at, is that can you change metabolism, and can that change PD-L1 with or without changing EMT as well? So those sort of peripheral, peripheral not in the context of reduced importance, but just in the limited geometric sense that I'm organizing them in my head, can that change as well? So what changes along one axis can propagate to which other changes, and to what extent on the other axis is something that is still we are working on.
Thanks.
Does that answer your question?
It does sort of, which is basically you're just focusing on EMT as the kind of like your metric, so to speak.
Yes.
and then seeing how all these other perturbations or aspects impact that. But you know, thinking about cancer and in particular, you know, metabolism, we know that tumors are generally more glycolytic, and we also know that a byproduct of that glycolytic metabolism is acidity, and acidity is bad for immune cells, right? So it's like immune suppressive. So there's this horrible sort of complex feedback between what the cell's doing, what the context is doing, and the dialogue between the two. And by looking at it through the lens of EMT, it's showing that there's even more sophistication than we thought. So as always, it's more complex than we first thought.
That's true, right.
Okay.
And now once we have the reason we are sort of stuck currently is because we don't have an EMT metabolism regulatory network yet. That's something we are working on. For PD-L1 we have one, and for drug resistance we have one in the context of ER positive breast cancer. So now we have coupled ER positive breast cancer, which I'll talk about now, and PD-L1 together, and there we see that when cells gain resistance to tamoxifen, they also upregulate PD-L1. And there is experimental data which sort of supports that hypothesis. So these two axes we have been able to connect reasonably to some extent, but metabolism is still, we are sort of lagging behind. Thanks. So yeah, so this is the third axis that I wanted to talk about, the ER positive breast cancer patients, which form the most dominant subpopulation of breast cancer cases. And tamoxifen was the first target therapy that was developed, which is capable of shutting down the estrogen receptor response. So the questions we were interested in asking is that, again, does EMT drive resistance, or does resistance drive EMT, or both? And what about hybrid cells? Are they also sufficiently resistant, similar to what we saw in the case of PD-L1? So we put together a network based on regulatory interactions known in the literature, simulated that over a large number of parameter sets in the ODEs. And what we see is that if you look at the EMT-ness, or you calculate EMT score, ZEB minus 200, you do see 3 peaks corresponding to epithelial-mesenchymal and hybrid. And if you calculate resistance score, you can clearly see a bimodal distribution as well. Now, when you put them together, what you see is that the most dominant phenotypes, each dot here is in silico cell in this case. The most dominant ones are epithelial sensitive and mesenchymal resistant, but hybrid cells are also capable of exhibiting resistance to tamoxifen, and they need not always, because there is hybrid sensitive as well. This is an interesting part which we are still investigating further. How can cells not undergo EMT but still gain resistance? Because you don't see on the other side that they undergo EMT but not yet gain resistance. So there is an asymmetry in the process which we are still trying to understand better. So we then did perturbations in our model and said, what happens if I overexpress ZEB1? So this is the model prediction. It says that mesenchymal resistance goes up. Then we looked at the experimental data where EMT was induced by overexpressing one of these molecules. So as expected, EMT profile goes up, EMT is more enriched, and estrogen response is down. And if you see this in multiple data sets, then you can do the other way around. You can change the resistance and see whether EMT was induced. This is what we see in simulations and in multiple different data sets as well, that when you silence estrogen receptor, which is what tamoxifen does, then you see EMT goes up. And when you generate resistant cell lines by constantly giving tamoxifen at higher and higher concentrations, again, you induce EMT. So it's a bidirectional switch. When cells undergo E to M, they undergo sensitive resistance. And when you push them to be resistant without inducing EMT, because of the circuitry, EMT is sort of automatically induced in this case. So once we had this, then we wanted to sort of look at a more population dynamics kind of behavior. So we had a very, very simple setup here, that there are 2 cell populations, sensitive and resistant. They can switch back and forth with some rate. And then we have this resistance score, which sort of tells you the probability of survival in this case. So here, the reason we wanted to look at these aspects separately is because often in the literature, plasticity and heterogeneity are thought of synonymous, and they are not. So plasticity can give rise to heterogeneity, but heterogeneity can exist even without plasticity in some sense. So we wanted to ask, what is more important, plasticity or adaptability, switchability, or heterogeneity without plasticity. So we wanted to decouple these two axes. So what we see here in these population simulations, when there is no plasticity, and all you have is varying levels of heterogeneity, and you treat them with a drug, eventually the population will collapse. So what heterogeneity can do is delay the extinction, but not prevent the extinction itself. What plasticity can do is expand a population even in presence of a drug, which heterogeneity was not able to do by itself. So then, broadly speaking, we saw these three different kinds of scenarios. One, when the population collapses. The other, when the population grows, similar to what you see here in these particular cases, when the population is increasing as a function of time, even in presence of the drug. And the other case where the population sort of stays critical, similar to where it started from, perhaps based on some dynamic equilibrium that is being maintained. So now we know from our previous simulations that tamoxifen.
Can I keep pestering you with questions? Sorry.
No, please.
I'm intrigued about this idea that kind of this, MET and resistance are sort of interchangeable in some sense. But we probably understand the mechanism of resistance to tamoxifen, like, at a molecular level, we know why it does it. I don't know from the cells you're looking at why it is, but I guess is tamoxifen like a standard sort of chemo? So there's probably some sort of pump sort of approach that's driving resistance. Do you know what resistance mechanism is being exploited?
Yes. So one of them that we know in this particular case is, in this scenario, alpha 66, this is the canonical isoform of estrogen receptor alpha, which tamoxifen is able to bind to and shut down the downstream signaling. Now, what has been seen in tamoxifen resistant samples, patient samples, is that there's an alternative splicing apparently that is happening up in addition to other changes as well in the ER processing aspect. That another isoform, which is not really seen in tamoxifen sensitive cells or at the parental population without treatment, comes up. And this has, as you can see, a different connection to ZEP1. So this one, which shows up under stress, activates EMT. But the other one, which was repressing this, is indirectly repressing EMT. So the two different isoforms itself, their relative ratio is triggering a more epithelial versus a more mesenchymal response. So the canonical form is constantly repressing EMT. It's repressing slug. It's repressing indirectly ZEP1, but this one is actually activating it. So it's basically those proportions which are controlling whether the cell will be more epithelial or mesenchymal.
So it's an anti-hormonal, so it's not a straight chemo, it operates on a different route.
Yes.
And so basically the resistant cells, do you think it's in order to get to resistance, they kind of exploit EMT in some sense, or or is it the other way around? Resistance drives EMT.
So that's something we are still checking in vitro. So and that's what my sort of last point in this population dynamics was, that what we are suggesting is that because the drug treatment itself can induce EMT, what we are proposing is that tamoxifen should be given together with MET inducers, so that MET inducers can push the cells back to being tamoxifen sensitive, which then tamoxifen can take advantage of. So those experiments are still ongoing to check whether these predictions make sense, and whether which one should be given earlier versus simultaneously. Those are questions we are still working on. But that's the combination we are suggesting based on the drug-induced plasticity itself.
Right.
Does that answer your question, Sandy?
It does, but it just makes me want to ask more, but I'm going to wait till the end.
Sure. So I just have a few slides, just one last thing I want to talk about. So in general, what we saw was that hybrid EM cells, now if you begin to characterize other axes of plasticity, have high levels of PTL1, do show reasonable resistance to tamoxifen as well in ER-positive breast cancer, and show higher glycolysis too. So what we are suggesting is that they're using these kinds of models, one can think about combinatorial strategies which can combat the drug-induced plasticity itself. I'll quickly rush through this part. I know I'm running out of time. Why are hybrid cells more plastic than the epithelial and mesenchymal ones itself? So here, what we did was we looked at multiple different networks that people have published in the context of EMG. And this is a particular representation here, which I'll get to in a minute why we have represented the networks the way they are. These are 5 independent networks built by different groups. So what you see here in terms of nodes, the red nodes are epithelial, the blue nodes are mesenchymal. The gray nodes are just inputs or outputs. So they are either just feeding into the system or feeding out of the system. They don't have feedback loops involved. The green edges are activation and the red edges are inhibition. So what you can appreciate, at least in some contexts, is that, let's say here, if you look at connections going from one mesenchymal to another mesenchymal, they are all activating in nature, or mostly activating in nature. But if you look at connections going on from an epithelial to a mesenchymal node, they are usually inhibiting in nature. So that suggests that although these networks look complicated, but they have a very simple principle underlying, which is that if you belong to an epithelial team, your goal is to support other epithelial players. If you belong to a mesenchymal team, you support members of the same team, and then you play a tug of war in some sense. So what we did here is to sort of make that point more quantitatively. We first had this adjacency matrix where we basically, what this tells you is that, an edge that begins from, say, GSC and goes to FOXC2. So this is the source, this is the target, and the color tells you whether it's an activation or an inhibition. But as you can see, this matrix is very sparse. So then what we said is that if you want to look at the influence of node 1 on node 2, it need not only be governed by a direct edge, but it will also be governed by various indirect edge paths that it can take. So then we basically multiplied this adjacency matrix by itself, or in other words, we took any 2 nodes in this network and took all different paths that go from node 1 to node 2, and then took a weighted average, the weight being the path. The longer the path, the less the weight. When you do that, you begin to see an interesting pattern that all epithelial nodes activate each other effectively. So this was done over a maximum path length of 10. All mesenchymal nodes effectively activate each other. All mesenchymal nodes inhibit all epithelial nodes, and all epithelial nodes inhibit all mesenchymal nodes. So now, instead of ZEB and MIR200, you have a family which is all ZEB and a family which is all MIR200. They are all inhibiting each other. So this was very intriguing to us. And then we wanted to ask that, what about this structure is so special? Is this specific to this biological network? So what we did was, we took our biological network, this one that shown here, and generated multiple non-biological, hypothetical, random networks in which, just as you would shuffle a deck of cards, you shuffle a set of edges. So when you shuffle these edges, you have broken the topological uniqueness. Now you have a different network. And in each of these networks, we calculated this influence matrix. Now, the important thing to note is this you can calculate without doing any simulations. So without doing simulations, just looking at the file itself, the topology file itself, you can say whether there are teams of players or not. Lo and behold, in the random networks, you do observe that the teams are broken significantly. So the teams are specific to biological networks as shown by this red line here. So it lies in 95th percentile or higher. There are some random networks which have stronger teams than the wild type biological network itself, but that's a small percentage. So then we further did analysis on this and we simulated this. So far there were no simulations. Now when we simulate and then you ask which steady state, which phenotype is how much frustrated. What does frustration mean? Frustration means that how many rules is it not following? So let's say if A activates B, if A is on and B is on, it's a non-frustrated state because the state follows the rule. But if A is on and B is off, then this state does not follow this rule. So it's a frustrated state. So you can quantify this frustration. Interestingly, across all 5 networks, we see a bimodal distribution of frustration. And the higher the frustration, the lower the frequency. The other characteristic we looked at is coherence, which basically tells you that if you perturb a network, whether it comes back to if you perturb a state, whether it comes back to the same state or not. Again, across 5 different networks, you see a bimodal distribution. And the higher the coherence, which means the higher the robustness, the higher the coming back to it, the lower the frustration. But this does not tell anything about which states are more frustrated. Where is the epithelial state here? Where is the mesenchymal state here? Where is the hybrid state? So then we incorporated that information as well. And the result was very clear. If you're looking at epithelial or mesenchymal states, they are much more coherent, much less frustrated, and have much higher frequency. So the epithelial and mesenchymal correspond to 95% of all the solutions that you see. The hybrid correspond to only 5% of the solution. Also, there is one way to define epithelial, one way to define mesenchymal, but many, many ways to define hybrid. So again, there is no one definition of hybrid itself. So it's like saying that all happy families are alike, all epithelial cells and mesenchymal cells are alike, but every unhappy hybrid state is unhappy in its own way because there's no one hybrid state, and there are many, many hybrid states that can exist. Then we asked, is there any role of teams in this particular case? So what we did in this case is now, instead of perturbing one thing at a time, we are perturbing many, many nodes, increasing increasing levels of perturbation. This is the behavior when you perturb epithelial, when you perturb mesenchymal, when you perturb hybrid. And this is showing coherence. What you see here is a very clear pattern that when you are perturbing terminal, which means epithelial or mesenchymal state, you need approximately 25% of the nodes to be perturbed to begin to see a difference. So if you change 25% nodes in your network, an epithelial cell stays resilient. It stays epithelial. A mesenchymal cell stays mesenchymal. But if you perturb only 5% of the edges, that is sufficient for the hybrid to push it out. And that's what we sort of further quantified this. That here you see a much more sigmoidal kind of behavior, that there's initial resistance when you perturb the system, and this is the change in phenotype on the y-axis. Then there's a sharp increase, and then there's a saturation. But if you do this for a random network, which does not have teams, you see a much more linear response. So this initial resistance is completely gone. So what we are saying here is that the presence of teams is actually enabling 2 deep phenotypes, 2 deep attractors, epithelial and mesenchymal, and a shallow set of attractors which correspond to hybrid. That is why you see hybrid can switch back and forth. But if you have weak teams, then you don't have these deep attractors. All you have is many, many, many different types of hybrid cells. And our current hypothesis is that this is what is happening when you're moving from development to cancer. In development, things are much strongly regulated. You have strong teams. But when you move towards cancer, the team strength goes down, plasticity goes up, heterogeneity goes up, aggressiveness goes up. That is something we're still looking into clinical data to try to validate. And this idea of teams is not only true for EMG. We looked at another network corresponding to small cell lung cancer. It looks a very complicated network, 33 nodes, 370 edges. You can reduce this network just to a 4-node, 10-edge system because there is so much redundancy which is present into the system. You can actually collapse many, many edges onto just one without losing much information as far as the phenotypes are concerned. And this is what we showed. Whether you simulate this system or this system, you get the same 4 phenotypes with comparable frequency. So this is all simulation. What about experimental validation? So we took these 33 nodes here and then did pairwise correlation, one with each other. So you can clearly see there is this 22 nodes which are all positively correlated with each other. These 11 nodes are positively correlated with each other. And these 22 and these 11 are negatively connected to one another, suggesting that there is one team here, the other team here, and they are mutually inhibiting each other. And that's what we see in experimental data as well, largely speaking. This is a cohort of cell lines, 50 cell lines corresponding to small cell lung. Another example, melanoma, where there are 5 different phenotypes at least that we know of, and we tried to construct a network which can possibly explain these phenotypes. we still see the same team's behavior. This is what the model predicts, that if you look at these 12 players, this forms one team corresponding to a proliferative phenotype. This forms another team corresponding to an invasive phenotype, and they inhibit each other. And this is what was seen in multiple data sets that we have. So here, we were able to sort of classify not only just broad proliferative and invasive, but also sub-states that can emerge when once these teams sort of begin to break down and so on. So I'll just summarize here that in multiple different networks that we have looked at of EMT, of lung cancer, of melanoma, we do see multi-stability as a recurring theme in dynamics of those networks. And once there are multiple states, they can possibly explain why you see phenotypic heterogeneity in vitro as well as in vivo, even if you begin from a seemingly genetically identical population, because heterogeneity can exist at genetic level as well, of course. Then the hybrid phenotypes we have seen show multiple features of high fitness. They are capable of evading the immune system by PD-L1 levels, capable of showing resistance to tamoxifen, for instance, and of course, because they are the most, they don't have any team structure supporting them, they are most plastic and least stable as well. And the networks which look seemingly complicated can have a simple underlying principle if one sort of looks more closely, and this has been seen in 3 different scenarios that we looked at here. And those nodes then shape the underlying landscapes. You can actually construct these landscapes in a much more accurate manner. With that, I would like to thank the fantastic team members who I've been fortunate to work with and presented some of the work done by various different members that you saw here. Thank you so much for your attention. I'm sorry for shooting over time, and if there is time, I'll be very happy to take any additional questions as well.
Nice. Thank you very much. This is quite a tour de force, and you have quite a team over there. It's a very large group you got. I warn you that people might ask questions in the middle of your presentation. I didn't know that would be mostly Sandy, but and I know that he has more questions. But before we move back to him, I don't know. I mean, this is definitely a topic that a lot of people around here are thinking about. So I don't know if other people might have questions now. Mishai? If not, I guess Sandy.
Heiko has a hand raised, or somebody has a hand raised. I think it's Heiko.
Oh, yes, right. There's 2 people on the list. Yeah.
Oh, hey, Mohit. Thank you so much. That was really beautiful. Sorry, for some reason I can't put my video on, but I had a question for some of the earlier part of your work that goes back to your PNAS in 2013, I guess, and probably throughout your work since, is how sensitive are these hybrid stages on the parameters of your model? So my question is, if you were to change some of the model parameters, would these switches and hybrid states completely disappear, or are they always there independent of parameter decisions?
Thanks very much, Heiko. I think that's a very important question. So initially, we did sensitivity analysis where we drew those bifurcation diagrams by varying 1 parameter at a time, plus minus 20%, and we quantified the range of the bifurcation diagram which corresponded to a hybrid phenotype. So there were certain parameters to which the width of that region was highly sensitive, but most of them showed a less than linear response in terms of their sensitivity. So that tells us that their existence is a more generic feature rather than, which is more robust. Now that we have looked at much larger networks, our confidence in that result has grown even further, because the definition of hybrid state can change, but they cannot be completely eliminated based on how things are organized and how nodes are influencing each other. So which hybrid state exists versus which one disappears as you alter a specific node or an edge is something we are still looking into. But the hybrid states themselves do seem to exist in a large parameter ensemble.
And so that's very interesting. Thank you. So is the prevalence of states in different cancers related to some of the parameters of different cancers, or just of structure?
Sorry, can you please repeat? I lost you, Heiko.
Yeah, so if you have different cancers, let's say breast, I assume you have different prevalence of hybrid states, different cancers. Yes. And are those different? Could those be deduced to different structures or due to different parameters in the intrinsic dynamics?
I think it's a mix of both. So in clinics, for instance, in clinical observations, hybrid cells have been seen to vary anywhere from 5 to 15%. So it's a small percentage, relatively speaking. And even there have been reports that if the number is greater than 3% or so, then that is sufficient to give a first survival. So their high prevalence might not be as dangerous as compared to just their existence in the first place as long. So it's more of a low pass filter than a high pass filter.
All right. Thanks, Mohit. Very, very nice work.
Thank you so much, Heiko.
So I know other two people who have raised their hands on Zoom to ask questions, but they've been lowered now. So I don't know if Heiko just stole the question or probably they just changed their minds. But not Sandy?
Do you have any, David?
Yes, I do.
Oh no, there's somebody else here. We should.
Mukherjee?
Yes, hi. At some point in the talk, you suggested that if we are breaking the feedback loops between in EMT, MET, it increases the metastasis.
It decreases metastasis.
Okay, I'm sorry. Then the question was more. Okay, I heard you wrong.
So one of the questions I had, and is maybe a bit more general, is obviously it's very clear from your presentation that plasticity and EMT have a huge amount of impact on not one thing, but also several of the hallmarks of cancer. I think that's what makes it so important. We think of the hallmarks of cancer, these separate things, but clearly there's one thing that underpins all of that. The way we apply treatment in the clinic for most treatments is, and this is one of the things that people at Mohit are trying to challenge, is that you just apply a drug until it fails and you apply something else. Do you think that in combination with what you found, we should be doing something different?
Yes, so that's the critical question.
It's a speculation, I know that, so don't worry too much about,
So as also I think Sandy was referring to earlier, right? I don't think we have a comprehensive map of what can induce what. We are just beginning to identify what axis can change which other axis. So it is possible that, let's say, tamoxifen and MET inducer seem a good combination as far as only these two axes are concerned. But when you bring in third axis, then things change in an undesirable manner. So once we have, so my hypothesis is that the reason cancer cells move together as a cluster, or a reason, not the reason, is that not a single cell is able to do everything and potentially cooperating. Now, this remains to be tested using this data. So can we reduce that heterogeneity, or if we push those cells all into being more like each other than unlike each other, which are then cooperating potentially, then that is one way to control aggression. That's what currently we are thinking about now. How to do it and which direction to push it is something we don't have a handle on.
So basically to reduce the heterogeneity, but not necessarily at the genetic level, but also at the epigenetic level, right?
Yes.
Very cool. Yes, I agree.
Thanks.
So any more questions?
I have a follow-up to that. So like David, that was sort of related to one of the questions I was going to ask. But it made me think about it even more, which is in a way, these anti-hormonals are quite a distinct class of drugs from, say, chemos or targeted drugs. And hormone regulation of cell state is a normal phenomenon in the body, right? That's like it's part of development, it's part of a monthly cycle, many processes. And so it's kind of intriguing then that you found that potentially inhibiting MET could make you more receptive or susceptible to an anti-hormonal drug. Right? If I'm understanding your results correctly. And that then makes me think about evolutionary therapies. right? So potentially, is there then a benefit to sort of priming with one and then inhibiting with the other? Could one cycle between these and sort of maximize control or maximize kill? Have you thought about any of those sorts of ideas?
That's a great point, Sandy. So we have not thought about in the context of tamoxifen yet. We have a more generic model where we are trying to see how plasticity, we are again trying to deconvolute the roles of plasticity and heterogeneity. And so far, there is no cooperation in our model. So that can add another layer of complexity. But we are trying to see whether plasticity and heterogeneity, how do they impact the efficacy of adaptive therapies? We have not yet gotten too much into detail of evolutionary therapies to the extent that you have looked into. And there, the model that we are looking at is in prostate cancer, because that's where, of course, most well-studied some of these ideas are clinically. So EMT also can drive resistance to androgen deprivation therapy. And androgen deprivation therapy can drive EMT or neuroendocrine transition, neuroendocrine prostate cancer, and so on. So once we have it in a reasonable format, I'll be happy to send it to you and get your thoughts. But that's where we are headed right now.
Yeah, no, I'd love to see that because it makes a lot of sense, right? And of course, it's going to be a horrible, big, complex feedback. But you know, on that note, because obviously prostate in particular is a male-specific disease, and you were talking about breast cancer, which can be female or male. But if Kristen Swanson was here right now, she'd be saying, is there any difference between male and female sort of MET states? And in particular, you know, is, say, that intermediate state more likely to appear in women than men? Or, because I'm sure you've heard of this idea of, you know, in some sense, at least in many cancers, they're seeing distinct behaviors in the cells from men and women. Like for GBMs is a particularly good example. But the idea that somehow, you know, male cells are going to stand and fight and female cells will kind of run and live another day, right? So that does relate in some sense to this transition, right? In a kind of crude way. I don't know if you've ever looked at that or thought about that.
Not in much detail. I mean, I would have to first look up which say cell lines are corresponding to say male breast cancer, and then we can categorize. I mean, the most ones that I'm aware of are from female patients, but that can be a starting point where we can see what the intrinsic EMT status of those cells is vis-a-vis various different female patient-derived cell lines. But that's a great point. GBM is something we're just beginning to look into, but the sex or gender differences is something we have not yet covered up. So that's a great point. Thanks.
It's something that's become critical to the point that every NIH grant now has to justify the gender and at least address the question of the model organisms you're using, if you're using male or female, or if you're trying it in both, and why not if you're not trying it in both, right? So it's considered kind of like we should be doing this.
That's a great point.
Good.
Cool. All right. Well, thank you very much, Mohit, for this excellent presentation, and definitely look forward to the report that's coming from your group, as well as all the one that you published in the last few years. So that'll keep us busy for a bit.
Yeah.
Thank you so much, David. Thanks so much, Sandy. It's great.
Thanks.
Thank you.
And it's what, 11:30, almost 12 almost in India.
Yes,
so I guess
yes.
Thank you.
All right.
Good night.